Okay, so we are going to wait for 2 more minutes. Okay, so let's get started. Before we talk about where we left, so any questions that you might have? And also, I understand that I think on Thursday, so that means tomorrow. So you guys have this which CSIR exam?
Yes, sir.
Okay, and apparently you are not comfortable attending this hybrid mode in the lecture hall, right? Is that true or what?
Yes, sir. I mean, considering that few students are giving the exam, yeah. So,
Okay, so then for this week, so we will continue with this whatever we are doing, and from next week onwards, so we'll think about shifting. Okay, it can be on Monday, but you would be updated whether this is going to be in a hybrid mode on Monday or not. But of course, on Thursday, because what actually we want to do is we really do not want to cover something new. Okay, so we just want to discuss what we already have covered in the very first class. Okay, because I don't know about the connectivity and so on and so forth. So many things are going to play out. So it could be on Monday or Thursday. Okay, so that will discuss and will come up with. Right. So keep checking on your WhatsApp group. Now, how many? What percentage of students now actually are on campus? So, Tanvi, you have a question? No, don't raise hand. Don't raise hand. So what I'm asking is, what percentage of students actually is on campus? Is it 50? Is it 90? Is it 80? Approximately. Vishnu,
So,
you have an idea?
so it might be, sir, I think around 60 or yeah, 65%, but I'm not sure, sir, again, because consider a few students are actually coming to and fro, so I'm not sure.
Okay. Well, see, I understand this is a theory course, and it can still continue as an online thing, but I'm a firm believer that the best learning can happen in an offline mode. Doesn't matter what kind of course you have. Of course, for practical, it has to be offline, but for theory, so if you are feeling discomfort in this online mode, obviously we have to shift it to the hybrid or offline mode. Okay, and as I said, maybe in the beginning, so we can have 3 classes where we cover the content in the online mode, and 1 class where we actually try to resolve your problems, solve any issues that you have, right, in the offline or the hybrid mode. So I think maybe for 1 or 2 weeks, so we can go this way, and then if, for example, almost all of you are here, and I think that probably for your second mid-sem you might have to be here. I don't know. So have you been communicated anything regarding that or what?
Not yet, sir.
Okay. Well, you discuss among yourself and let me know what suits you the best. Okay. And that is what we are going to do. But my suggestion is, at least for initial 1 or 2 classes, that means 1 or 2 weeks. So we do continue in the same mode, but one of the classes where we have discussion, tutorial kind of thing, resolve your issues, that can be done in a hybrid mode. Right. And if you are here, so you come to the lecture hall complex and you can raise your questions, and same is the case with whosoever is joining online, right? And let's see how it goes. Right. But over the weekend, so if you have a group on which you can discuss, so discuss it and maybe come to a consensus. All right, so question on what we covered. Well, if not, so let's continue. Okay, so you're talking about these antigenic determinants that are present actually in the antibody molecule. And these are three antigenic determinant. One, we call them as isotypic determinants. Second is basically the allotypic determinants, and third one is idiotypic determinants, right? So this talks about isotypic determinants, which are actually present in the constant region of heavy and light chain. Okay. For example, the isotype or the subclasses, the classes of antibodies that you have, IgG and IgM, so they will have invariably different isotypic determinants, because these are actually present in the heavy chain constant region. Okay, let's say now if you assume that you are utilizing the same light chain, kappa here, kappa here, so then basically those determinants are going to be same for the light chain. All right. The next one was the allotypic determinant, where actually you are talking about two different strains of animals. Okay, but you are talking still about the same antibody molecule, like the same class, IgG1, for example, right? Largely, so these are conserved, right? So you have basically this and this same, this and this same, this and this same, this and this same. But some of these epitopes might be uncommon between these two, and that can show some level of reactivity. For example, if you generated antibody against this antibody, so that actually will recognize this epitope, it may not recognize this epitope, right? This can also vary, for example, in the light chain concentration. Okay. One is constant, for example, and another is different. Okay. So the only difference is the strain of animals are different. Or in other words, if you talk about humans, so one individual versus the other individual. So that's what we are talking about here, right? Third class is idiotypic determinants, which are actually present in the variable region, right? So this is the variable region for both heavy and light chain, right? And as you can see, this is because of the unique amino acid sequence that you have in the V region. And that is why for any antibody that you want to use it for clinical usage or for other purposes, so it has to be checked whether the antibody actually is showing immunogenicity or not. Okay, even if it's coming from the same animal, same species, so that also has to be tested for the antigenicity. And some of the determinants which are actually present in the variable region can be different, and they might induce some reactions. Okay, so the other part of the statement actually is, in some cases, an idiotype. Okay, so we call it as an idiotype or idiotopes. Idiotope, basically, you can think of it as epitope, paratope, idiotope, right? It has nothing to do with idiot. Okay, so it's idiotope. So this is how you call it. And idiotype is the actual binding site. So that means you have this antibody molecule, right? So let's consider this is one paratope where your antigen will come and bind. Okay, so this paratope actually can also serve as the idiotope. Okay, so this is what it means. And it has a significance even in fine tuning your immune responses. Rather, one of the most popular theory when proposed for antibody formation and immune regulation was this immune network theory given by Neil Young. The other name of this theory is idiotype anti-idiotype theory. Okay. So for example, if you have generated antibody against this part, so this will bind to this, right? And whatever the antibody you have generated, that also might show some unique antigenic determinants against which you are going to actually then generate antibodies. But you have to remember, because these things are present in the variable region, so invariably, these are very, very poor in immunogenicity. You actually have to come up with very fancy techniques to even show that this might exist. Okay, theoretically it might. You can say that, yeah, there is a possibility of having anti-idiotypic antibody and so on and so forth, and its role in immunoregulation and the subsequent processes, right? But if you actually want to show it, maybe 1 in 1 million or 1 in 1 billion kind of cases we are talking about here. Okay. This is not to say that it doesn't have any value. Rather, there is one antibody which actually is an anti-idiotype antibody, which actually is in clinical trial for shutting down autoimmune reactions. Okay, so this also has a practical utility. But understand this, the idiotypic determinants are present in the variable region, allotypic and isotypic determinants are present in the constant region. And if you basically grade based on the antigenicity, okay, so the level of immune reactogenicity, right? So it will be isotypic determinant first, then allotypic, then idiotypic. Okay, so this is the gradation. Okay, so although we will discuss signaling through BCR at later time point as well, but this slide actually highlights the
Excuse me, sir.
yeah, sure.
The gradation you told, this was about of what?
The amount or the number of epitopes you can generate in antibody molecules. So predominantly, these are present in the constant region, and they are classified as isotypic determinants. Okay, the next immunogenic, so you understand it this way. So for example, if you inject an antibody in a different species, so you are going to generate for sure anti-antibodies. Okay, and those anti-antibodies are predominantly present in the constant region. Okay, if you inject antibody from one individual to the other individual, still you can generate immune responses. These are less abundant. Okay, and the recognition is predominantly in the constant region. Okay, and third one is basically from your system within. Okay, so you take out your antibody and other antibody clone. So basically within your system, so you might have anti-idiotypic antibodies. So you generated antibody against one antigen, for example, right? And against this antibody that was generated, that was actually found by your B cells, some other B cells of your can also recognize epitopes present in the variable region. Is that clear?
Yes, sir.
Yeah. B cell receptor. Okay, so you know that this actually is a membrane-bound immunoglobulin, right? It has this variable region consisting of both heavy and light chain. So this is, for example, heavy chain, this is light chain, variable region, constitute a paratope, this can bind to antigen. But on the other side, this is also tethered to the membrane, to the plasma membrane. Okay. And in order to activate your B cells, the antigen has to come bind to this variable region, right? And it stimulates signaling through a BCR, right? And then this B cell, which has actually seen this antigen through this membrane-bound immunoglobulin, can get activated. Okay. So essentially, membrane-bound immunoglobulin would have a transmembrane region as well as a very short cytoplasmic tail. Secreted antibody will not have this transmembrane and the cytoplasmic tail. So it will be only up to this point. So basically, how this is happening, this is not like you come up with this antigen which bind to here, and with this recognition, so you actually process it here right away on the surface. No. So this actually requires a lot of signaling transduced within these B cells, right? And because of the signaling, whatever the new molecules you are going to produce, so they will be having the same antigen specificity, but they will be processed antibodies in the sense they will not have transmembrane and the cytosolic domains or the tail, right? Now, one thing you would appreciate from this cartoon is that there is actually very short sequence of amino acid or very short tail, which is actually projecting in the cytosol, in the cytoplasm, right? And this is not sufficient for transducing signal within these cells. Even if you actually cross-ligate, either the cross-ligate these BCR on surface of your B cells, or you have the antigen binding to it, right? Now, in order to get away with this problem that this short cytoplasmic tail is present in the basically immunoglobulin molecule, which is membrane bound and serving as the receptor. So you have accessory molecules, okay, which serve as accessory molecule to the B cell receptor. Okay, in case of BCR, we call them at Ig alpha and Ig beta, right? So they do not have a major function which is actually present in the extracellular region, except for movement of the chains within the lipid raft and within the membrane itself, so that as this antigen actually is sensed by your BCR, so these other molecules kind of move around in the membrane. So they actually have this lateral movement in the membrane, right? And in addition to that, it has actually a transmembrane region. Obviously, it has to, because this is actually responsible for signaling, and a very long cytoplasmic tail. So 48 amino acid, for example, in beta, and 61 amino acid in alpha chain. And why this is critical to have a longer cytoplasmic tail? Because for signal transduction to occur, so you require particular kind of amino acids present in the cytoplasmic tail. Particularly tyrosine has to be there. And tyrosine has to be phosphorylated, for example, in order to create these docking sites to which other molecules can come and bind and then initiate this cascade of events, right? And one more thing is, for example, this BCR present on one cell will have one antigen specificity caused by this variable region in the constant and variable region in the heavy and light chain, right? Other B cell clone would have it different. So it will have a different antigen specificity. All other things will remain same, right? But so what is happening here? Why people keep on logging on this side? Anyways, the same accessory molecules like Ig alpha and Ig beta, so they actually can serve as the component of B cell receptor, right? When you actually recognize a different antigen. So it can be the same molecule. So this basically can be utilized by several different types of BCRs present on surface of different types of B cells. Not exactly. So obviously this molecule also has to be present in the same cell. Okay, but you don't have to diversify this. This remains more or less constant. So here you have to diversify, particularly in the variable region, to serve as the B cell receptor, so that it can recognize either one antigen or 100th antigen or 1 millionth antigen or 1 billionth antigen, right? So this is what essentially it means, that you require accessory molecules to transduce signaling in the cells which are recognizing antigen through their BCR. Okay, so the other thing that we briefly did mention in actually at several times, one of them was actually this poly-Ig receptor, right? These are some of the key molecules, crucial molecules that also are part of immunoglobulin superfamily, but very critical in different biological processes. And they have to be actually studied in context with antibody molecule, because they are actually part of the immunoglobulin superfamily. Okay, these are Fc receptors, and they actually can bind to Fc region of the antibody. Okay, so all these. And now you can appreciate that there are several kinds of those. One of them actually is this poly-IgR, which is actually responsible for transcytosis of your antibodies from, let's say, the submucosal site in gut to the lumen side, where microbes are present, and basically then this antibody, after migrating into lumen, can neutralize these microbes, right? The other Fc receptors that you have are, one of them actually is called as Fc receptor N. N stands for neonates. This actually is present in neonates, right? And this actually is very critical for actually transport of some of these antibodies even across placenta, right? IgG antibody can also bind to it. Now, you will learn in the subsequent slides that this actually is very similar to MHC class 1 molecule, the structure of this. Okay, so you have 3 domains, beta 2 microglobulin, but it really does not present any antigenic peptide on its surface. Okay, it binds to the Fc receptor of antibody molecule, Fc receptor neonates. Okay, so these are 3 receptors which actually can bind to your immunoglobulin normally, and they have actually different roles to play. So some of those we'll discuss at some time point. Okay, you have Fc receptor gamma R1, gamma R2, and gamma R3. So these are basically CD nomenclatures: CD16, 32, 64, right? And now you can see that these Fc receptors, they are just not like they bind to the antibody and have to do nothing. Okay, so they bind to antibody and they can also transduce signal, because the accessory molecules that you have can transduce signal in these cells. You have the transmembrane portion, which can associate with these Fc receptors present in the membrane. So basically, this is transmembrane domain interacting with the transmembrane domain, and this is because of the positive and negative ions interactions, rather positively charged and negatively charged amino acid interaction within the membrane, right? So you have gamma, beta. So these are the accessory chains which actually are required for transducing signaling through Fc receptors, right? For IgA, so you have this receptor, Fc alpha receptor, and for IgE, which actually is responsible for causing these allergic reactions, so you have Fc epsilon receptor, and this actually is present in large abundance on mast cells and also on eosinophils. Okay. So these are the FC receptors. So they can bind to antibody. Antibody, if bound to this antigen or allergens, they can transduce signaling through this FC receptors and then carry out different functions. Okay, different function can actually range from internalization of this FC receptor, transport of this cargo which is bound to the antibody and internalized, and then it can be processed, for example, by your innate immune cells in certain way, and some of these peptides presented internalized and processed eventually can be displayed on MSC class 1 or class 2 to activate your T CD4 or CD8 T cells. So that also is these receptors are also involved in the process of antigen processing and presentation. Okay, particularly in B cells and also to some extent in macrophages, because they also have these receptors, right? And obviously for epsilon receptors, so you have these allergic responses. For example, if this is happening on surface of mast cells are going to release these granules, and these granules carry histamine. Histamine causes allergic reactions. So this receptor is critical for that. For example, if you can find an antibody against epsilon receptor, which prevent its binding to IgE molecule, so you can actually control allergic responses. Obviously, you can use antihistaminics, right? But this is one of the approach that you can see just by looking at these receptors and their biology and their physiological function or pathophysiological function. So you can actually come up with strategies to different approaches to manage different diseases, right? Okay, function of FC receptors. So we did talk about that. They are very crucial for biological functions, right, of antibody molecules. For example, complement activation was one of those things, right? But in FC region of the antibody molecule, so you have this complement binding site, right? And allergic responses, for example, transport of these antibody molecules from submucosal site to lumen. For all those things, you require several of those receptors. Okay. Obviously, for transport of your immunoglobulin to fetus across placenta, and these receptors can be bound to your antibodies in a passive way. For example, you have generated IgE kind of antibody in response to an allergic stimulation. And this process actually is called sensitization. You generated those antibodies can bind to these receptors. And when at a later time point, you get exposure to the same allergen, so you actually generate a very acute allergic reaction. Okay, so these are all the functions associated with these FC receptors. Any questions that you might have on this? Okay, if not, so briefly, we will talk about immunoglobulin superfamily members. Although this is just like an information that, for example, as an immunologist, you want to know. Okay, any protein can be classified as a member of immunoglobulin superfamily if it has this immunoglobulin fold. And we did talk about what immunoglobulin fold is. So you have this 60 to 70 amino acid loop, intra-chain disulfide bond, right? So if you have this structure in any protein, so that you can start calling as the member of immunoglobulin superfamily, right? We did talk about these Ig alpha and Ig beta, which actually are part of the B cell receptor, immunoglobulin, which actually is membrane bound or secreted, right? T cell receptor is also part of immunoglobulin superfamily. So this also has the same thing, right? MSC class 1 or class 2 molecules are also part of your immunoglobulin superfamily because they also have this immunoglobulin fold. Okay, beta 2 microglobulin. This also has the same thing. With beta 2 microglobulin, it actually associates with MSC class 1 heavy chain non-covalently. So it does not really form covalent bond with MSC class 1. Non-covalently, it interacts with it and then present those peptides which can stimulate your cytotoxic T cells. Okay. So you also have, yeah.
So what did you say when you said that by creating an anti-FC, like antibody against FC, we can have an acute allergic response?
Okay, so I think you're talking about this one here, right?
Yes, sir.
Or actually the function. So for example, what I'm saying is you exposed to the allergens, for example, pollen grains, dust, mites, okay? I don't know, cat fur. Some of you actually like to have pets, basically dendron, which actually is basically these protein which are coming from skin of these pets, dog, cats, hamsters, whatever, right? So these things can stimulate your B cells, and these B cells can then start producing IgE kind of antibody, right? Once those antibodies are produced, those antibodies can interact with particularly the Fc epsilon receptor, right? Even if you don't have the antigen, so these antibodies are passively present there. So they are actually there. They are already there because they can interact with high affinity with Fc epsilon receptor. Now, at a later time point, for example, if you get exposure to this allergen, okay, so you might be saying that I'm allergic to this thing, I'm allergic to the other thing, right? Peanuts, okay, some different kinds of fruit, pollens. So all these allergens, right? So you might have different allergies, right? So what I'm saying is, at some time point, for example, if you get exposed to that allergen, so you produce antibodies, those antibodies can bind to these Fc receptors and are sitting there. Okay, so this process actually is called sensitization. So you don't actually observe the effector functions, and eventually, so you don't have discomfort and all those kind of things. But at a later time point, if you actually get exposure to the same allergen, so this allergen is going to bind to this antibody, which actually is immobilized or bound to Fc receptor, right? So this actually can cross-link multiple Fc receptors, and as a result of it, so you transduce signaling in these cells on which this antibody is immobilized. And I also said that Fc epsilon receptor are predominantly present on surface of mast cells. Mast cells carry these granules which have a lot of this histamine. As a result of this interaction on surface of these cells with allergen, cross-linking of the receptor, you transduce signaling, and these preformed granules basically open up and histamine is released, and histamine carries out all the functions or making you sick or making you allergic, right? This is what I said about the allergic response and the role of this Fc epsilon receptor. Yes?
Okay, sir.
I also suggested that, for example, if you are allergic to so many things, so it's not a bad idea to block this FC receptor using an antibody therapy, which actually is not a signaling antibody, but a blocking antibody. So you don't actually have the constant region of antibody binding to it, but the variable region, so that your IgE does not interact with it. Okay, this is just one of those things that can be tried, and I'm pretty sure people must have tried it. Okay, immunoglobulin superfamily. So there are more molecules which are actually enlisted here. So all T cell accessory proteins like CD2, CD3, CD4, CD8. So all these are part of immunoglobulin superfamily. And the reason is because they have these domains in a particular configurations, a sequence of amino acid with intrachain disulfide bond. Okay, poly-IGR, adhesion molecules. So these are all part of immunoglobulin superfamily. Okay, you can remember if you want, but this is just to stress upon that it's a very large family. A lot of biologically relevant molecules actually are present in immunoglobulin superfamily. Not only immunologically relevant, but in many other functions as well, in many other biological processes as well. Okay, so the next topic we are going to talk about actually is monoclonal antibodies.
Excuse me, sir.
Yeah.
So what are adhesion molecules in the last slide?
Yeah, adhesion molecules are responsible for multiple functions, particularly in immune system. So, for example, you have to have this cell-to-cell interaction, right? And for cell-to-cell interaction to happen, so some of these molecules are expressed either on the surface of these cells or, for example, in the endothelial cells, right? And because of these interactions, these cells either can get engaged with each other, or they can move within the blood vessels, or they can actually get out of blood vessels and reach to the site where they are supposed to. For this whole process, you require these adhesion molecules. Okay, there are several kinds of adhesion molecules. ICAM is one. L-selectin, integrins. So several kind of molecules can be classified as adhesion molecules. Okay, they are highly glycosylated on the surface, so they are basically a lot of carbohydrates are present on these molecules. Okay, predominantly present in extracellular matrix. This is not only responsible for immunological function, but also during the developmental processes. Okay, if cells, for example, from one cell you have made multiple cells, and if these cells keep on hanging around everywhere, so you don't develop as an individual or as an organism, right? So you require this adhesion property, cell-to-cell interaction, right? Only then basically organogenesis is going to take place, and eventually you will form an individual, right? Something like that. So these molecules are very critical. If you can disrupt function of a very critical adhesion molecule, so chances are that you get all kind of inflammatory problems. You may not be actually even able to develop into a fully grown individual. So all those things are there.
Okay, sir.
So
And
the, yeah.
In the loops, we can see that C and V are written over there. So is this C means constant, V means variable?
Well, yeah, essentially this is what it means. And this actually means basically this region can actually change, and this remain constant. Okay. But this is not basically the kind of antibody molecule that you have. So where you have the hypervariable region and not so variable regions. Okay. But for example, some amino acids might be different here. But here, so more or less, all the amino acid will remain constant from cell to cell, from molecule to molecule, from individual to individual. This is what it means. So these are basically shown as the carbohydrate decorative side. For example, you might have asparagine here, so which can be end-linked glycosylation, right? All those things are there. Okay, so when I say monoclonal antibodies, so actually, what do you understand by that?
Antibodies, those bind to a special epitope, a specific kind of.
A specific epitope. And opposite of that would be?
Polyclonal.
Huh?
Polyclonal antibody.
Yeah, obviously, polyclonal antibody. Okay. So essentially, for example, if you get 5 ml of your blood, okay, and isolate antibodies from it, let's say IgG. So what kind of antibodies are these? Monoclonal or polyclonal?
This should be polyclonal.
Okay. Okay, let's say now you have actually isolated one particular B cell and you find a way of immortalizing this cell so that this remains forever and it keep on producing these antibodies. So this will be the antibody produced by this clone of a B cell.
Monoclonal.
Obviously, right? So this is what we are going to talk about, how to generate these monoclonal antibodies. Okay, so we already have talked about this slide. From stem cell, you have this gene rearrangement processes, and then you are going to generate these different clones of your B cells. Once they mature, they get into peripheral circulation, and once this antigen come and the BCR recognize that particular antigen, so it expand, right? And following expansion, so whatever the product this B cell will make, so they will have the one and the same antigen specificity, right? Now, what you actually have to do during generation of monoclonal antibodies is you have to find a way of selecting one of these clones and immortalize it, right? This is what is the goal. And that is what actually is shown here, the broad schematic, okay? So you have, for example, a mouse, which you have injected with this antigen. Antigen has multiple epitopes of different types, right? So that means following this injection, so you are going to generate antibodies producing cells, which can make antibody against epitope 1, epitope 2, epitope 3, epitope 4, right? So what you actually do is you have injected it and you follow a schematic that you keep on injecting this antigen 4 times so that you actually have enriched your splenic tissues or lymph node with significant number of cells which actually have B cells able to produce antibodies against these different epitopes, right? So in spleen, so you will have these B cells which can recognize and produce antibody against epitope 1, 2, 3, 4. Okay. So what you do is, these are called actually plasma cells because these are antibody producing cells, right? So you fuse these cells with myeloma cells. Okay, so myeloma cells, as you know, they are actually cancerous cells, right? Once you fuse these two cells, so basically this process is called hybridization. Okay, and that is why this process is called hybridoma technology. Okay, now after fusion, so what happens is, so the property of antibody producing is provided by the clone of B cells. Okay, and the property of immortalization of your B cell is provided by this cancerous cell. Okay, immortality is provided by your myeloma cells, and the ability to produce antibody is coming from your B cell. Okay, so that means if you actually can select these different clones, so this will remain forever, because now this is actually serving as a cancer cell, and this will keep on making antibodies of that one particular antigen specificity, right? For example, it is clone 1, so for this epitope, for clone 2, this epitope, and so on and so forth, right? Hybridize, you generate this hybridoma, and then you have to actually follow this very exhaustive, very extensive process of selecting these different clones, different hybridomas. Okay, so for example, if you are able to basically select only this kind of in a separate well or separate flask, so now whatever the antibodies are produced by this particular fusion product of these two cells will be these antibodies that will have specificity towards epitope 1. For this one, specificity towards epitope 2, 3, 4, right? Contrary to this, polyclonal antibody is, for example, if you isolate serum from this immunized mouse, in this serum, so you'll have all kind of antibodies. And it's not easy to isolate one antibody molecule which bind to this antigen versus this antigen versus this antibody. But cells can be isolated, and then they can be immortalized, and then you keep on making this antibody up to eternity. Okay, several hundred divisions. So this is actually the essence of the whole process of generating monoclonal antibody, right? Any question on this?
Sir,
Yeah.
so in the final process, how do we select these 4 antibodies specifically in the 4 tubes? So
You have to do a lot of screening, so I will talk about that. So this is called limiting dilution assay, LDA. So what you do is, so basically, let's say if you have generated these fused products, and you have counted the number of cells, and let's say you have 1 million cell in 1 ml of the medium, right? You dilute it in such a way that if you aliquot 100 microliter, that 100 microliter have from 0 to 1 cell. Okay. In some well, if you aliquot this 100 microliter, you won't get anything. In some, you might get 1. Okay. Chances are that you, in some you might get 2. But then you have to actually screen that you have actually selected a single cell. And because this is a cancer cell, in next 2 or 3 days or even 4 days, so this cell is going to divide, and it will make actually multiple copies, right?
fine, sir. Yeah.
Well, this is not a trivial situation, so but nonetheless, it was shown by Caesar, Milston, and rather these two scientists, this Collar and Milston, right? And obviously, because they devised this technology, which more or less revolutionized the whole field of biology, because you can generate these antibodies against different epitopes, and antibodies are used for almost every single biological discipline, right? And they were actually recognized for their contribution to develop this monoclonal antibodies through hybridoma technology with Nobel, right? So this is the process. Inject a mouse with a specific antigen to stimulate its immune cells to produce necessary antibodies. Extract mouse spleen containing B lymphocytes and culture them in the lab. Extract mouse tumor cells, or you can actually keep on propagating these cells, right? Tumor cells, this is the key, which grow continuously, and culture them in the lab. You mix spleen cells with tumor cells on the same plate and culture these and when you have added your cancer cell and you have added your B cells in the same plate, so you are going to get these fused products, right? You obviously you have to add some reagents, some chemicals, or maybe some other proteins which actually can carry out this fusion processes, right? Polyethylene glycol, for example. This causes some B lymphocytes to fuse with tumor cells to produce a hybrid cell. And this hybrid cell is called hybridoma, right? Now you grow these cells under conditions that allow only hybridoma cells to survive, right? Extract these cells, culture them separately to test the medium, because whatever the product they are going to produce, and in this case, it's going to be antibodies. So that antibody will be actually present in the culture supernatants. You collect culture supernatant and find out whether this is reacting to your epitope or not, or to your antigen or not, right? And culture the cell, making the desired amount of antibodies and use as needed. Okay, so these are all the processes. There's some question. Yeah, we'll get back to your question. So how do we avoid non-specific hybrids? Well, you cannot really avoid. Okay, so let's talk. I have actually made one slide where it will be more clear to you. Okay, so yeah, well, here you go. Okay, so you have fused these plasma cells with these myeloma cells, and these myeloma cells actually are deficient in these two enzymes, HGPRT, and obviously they are not supposed to produce any antibodies of their own, because otherwise it will defeat the purpose. Antibodies, whether they are coming from this cell or this cell, so you don't know, right? So they have to immunoglobulin IgM deficient, and IgM deficient means they cannot really make any antibody. And they also have to have disrupted function of this enzyme, HGPRT. What exactly does it do? So we'll talk about in subsequent slides. Okay. So you fuse. So these are different options that you're going to get. You will have unfused cells. And obviously, because these are primary cells which are coming from mouse, after some time they are going to die because of senescence. You might have homologous fusion of your primary cells, B cells, B cells. This also is going to die because of senescence. Okay. You have, for example, because these are deficient for this HGPRT, so you actually culture these cells in het medium. So what is the composition of het? So we'll talk about in subsequent slides. Okay. So, but consider that you have to actually grow these cells in a specific medium, which is going to kill the primary cells. All the primary cells will be killed. Het medium. So this is going to kill these cells. This is going to also kill basically the deficient cells. So basically, HGPRT deficient cells will not grow in this medium. Okay. Now you can say that this is also HGPRT deficient, right? But remember, so this now actually is present as a fused product with your primary cells, right? Now that HGPRT actually is coming from your primary cell. So this will rescue this particular hybridoma. It's not going to rescue these two things. Cancer cells actually do not like this medium, so they die when you culture them in het medium. Okay. These cells survive because of the Cellular pathway. So we'll talk about what Cellular pathway is. So obviously, because this is a fusion of two cells. One is cancerous, which is basically not going to get older, right? Or there won't be any senescence, which actually is a property of your primary cells, right? And they can utilize the Cellular pathway for selection. So now in this case, so you have only this thing selected. Broadly, is this clear? Not to mention that we have to talk about SGPRT, role of this SGPRT, immunoglobulin deficiency and het medium, and what is Selvage pathway and what is the other pathway. Okay, so all those things we are going to talk probably in either tomorrow's class or in subsequent classes, maybe tomorrow's class. But this is essentially the process is for generation of monoclonal antibodies. Yes, I think we have to now stop. It's already 9:59. Okay, we'll continue from here on.